See that the people who are connected is stable. So again, thank you for joining us for this first webinar of 2025. Very happy to gather all around the world. We are just coming back from two major events, CES at Las Vegas, where we had an innovation award that was tremendous, and GPM, and we would like to share with you the actual demo we made in Las Vegas. That was the first real life demo of the entire ecosystem of Netry. So let's begin. I'm gonna share my screen. Here you go. Yes. So perfect. So a little bit of a wrap up of what we do at Netry. Again, we just try to introduce our brand new neuron as a sensor approach to wrap up a little bit. So we basically galvanize the fact that all the organs are innervated by our peripheral nervous system that has been educated for the past 100,000 years by Darwin to develop a complex yet efficient way to gather all the biology that has been processed by the organs. As you can see, everything is connected through the peripheral system that goes to the spinal cord up to the CNS. And we use this way to see the neurons, not only as a cell per se, but as a sensor to digitize the human biology. So how does it work? Basically what happens is that we have at the endings of all those nerves termination, you have all receptors that are dedicated to one specific organs. Now that being said, each organ has specific receptors, and that integrates everything that happens into a specific organ at the nerve endings. By recording the electrical activity of those neurons over time, we can record baselines of the normal way that the organ works, but also respond to a compound that will interact with the organ and thus with the receptors that are included in this organ by innovation. By doing so, you are able to record this complex signal that is integrated through the PNS up to the CNS, so that we can actually make what we call a digital signature that is specific to a specific compound. And as an example, we have 6 metrics that we expose here with a response to inflammatory response compared to temperature. And we plot only 6 variables that we can measure with this neural activity integration. And it happens that we have up to 42 different variables, PNS and CNS, that describe this digital signature. So for the purpose of the next slide, I'm just going to represent those 42-dimensional-ish measurements by what we call a digital signature of a compound of a condition. And by making comparison with all the compounds, reference compound or compounds from customers that we have screened, we can build what we call a digital library, again in 42 dimensions, that allows us, with our machine learning system, to make the distance between the compounds that know to have failed in clinical trial or compound that are on market or reference compound to make a digital match to the compound that is tested. Therefore, you can use the digital signature to compare to the compounds of condition we have and use it to predict safety and efficacy or diagnose what is tested into preclinical trials or clinical trials. Now, that being said, we can use this methodology in the entire path of development, from target validation to pharmacology and safety, with the entire multi-electrode platform that I'm going to expose you just after. Now, we want to focus a little bit about what we have called the NeuroFluidics Care system, that is an entire pipeline of analysis using our devices, wetware, and software. So at Netry, we have developed devices that is composed of 16 different individual chips, as you can see, in a 96 well plate format. This is very important because we want to have a lot of data points, and those formats is completely automatable via high throughput platform that everybody has in their labs. You can see 96 alignment well plates. Now, when we zoom in into the active component of the device, you can see that we have created 3 different compartments. One compartment that we call spinal cord compartments, where we can place the cell body of peripheral nervous system or central nervous system that sprouts into another compartment when you call organs compartments. And the key is to use microchannels that are 3 micron height that allows only axons to sprout from one compartment to the other. This allows us to make a pure fluidic isolation, meaning that on the organ part, you can place either 2D or 3D cells of organs and have termination that of the nerves ending, and apply a drug only on the endings part, going back to the initial slide that I showed you, where you can target the interaction of the nerve termination with the organ by synapses or directly without any synapses. So we either can screen neurons per se for different application in pain, especially oncology, or neurons plus the supernatant that is given by an organ. For example, in the gut, you can directly look at the penetration of a component or microbiota into classical standard culture and take the supernatant that is underneath, or directly into our devices as a core culture. We have several types of devices available. 2D cell culture with different lengths of microchannels and channels for 2D culture, synaptic compartment culture with the directionality, or 3D or 3 culture, but also 3D cell culture system where you can innovate a biopsy, an explant, or 3D reconstructed organoids or spheroids. And we also have currently developing the duplex link MUA system, where you can not only innovate from the side with the blue compartment, but also vascularize with a micromembrane that is placed under the opening here and the microchannels that are underneath for constant perfusion and irrigation of the organs that is in the wet spots. But every platform is connected to our multi-electrode arrays capabilities to go back and use the neurons as a sensor. Now we have developed, and we are currently developing a full range of more complex platforms where you can create only barriers, as an example I give you with the skin, with our vascular skin. Also innovation and vascularization of several organs in a multiple organoid system connected by a jumper system, but also with our duplex link MEA, as I showed before, to not only connect multiple organs, but look at the influence of multiple innovation simultaneously without any pumping system. Now, we connect those platforms using our partner Axon Biosystem Maestros Pro or Edge to monitor the electrical activity of neurons and extract the electrical digital signature of different compounds. And for that, we have created softwares, NeuroFluidics and DataLink, that we are going actually to show you for the first time in this presentation. Just to come back on digital signature, we are able to apply compounds in a chronic way to look at the alteration of the states of different variables over time. That can be after 1 hour or after 24 hours, and we've been up to 7 days after application of compound. We also have the capacity to look at immediately in the fast response with an acute exposure to look at the difference of folds between the direct electrical response of a compound in all our platforms. As I mentioned, we can recall up to 42 different variables that describe the entire neural network, PNS and CNS, and we record the electrical activity through the entire channels and microchannels simultaneously. That will give us a full digital signature over time. We have the capacity to extract a lot of different parameters and to compare those parameters together using our uplink and new software implementation, where you can make a lot of graphs and comparison in a very user experiment oriented design. And again, Maestro, we use our extraction system that goes in storage into our cloud system. Now, how does it work directly? So we make always a difference of alter state between reference state, and we can just measure the difference on the entire variables and over time. We have also developed a quality control plot that allows us to look at the normalization of the spontaneous burst rate of the culture just before applying the compound. That allows us to, since there's a variability between batch to batch that tends to diminish with the quality of the cells, but we can, before application, assess the capacity of the chip and the cell culture to respond properly using this quality control software device. Now, how does it work? So I have a couple of videos I want to share with you that we recorded before. So the NeuroBento system that is here is seeded with cells in a very easy manner, and this is the way it works. This is a live presentation, so thank you Aurélie for and Camille for making this happening. You can see that cells can be placed. So we use colorant in this particular video to seed and the cells and maintain the cell culture flow, since you don't need any pumping because we use this PDMS pump-free system. And you can see it's easier as gravity driven to place the cells, maintain the, and to renew the cell culture media that is directly placed into the incubator over time. Every 3 or 4 days, the media is changed, and after a couple of weeks, we are able to record the electrical activity of neurons. And I just want to move on the next video. Also, the NeuroBento system can be manually tethered to a high throughput culture system, or doing by pipetting. And I wanted to show you in this quick video how in our facility, how can you maintain up to 110 NeuroBentos in the same time to make cell culture over 3 weeks. So those devices are totally compatible with high throughput automation system for cell culture maintenance, but also for imaging system and eventually MEA multi-electrode arrays. And you can see everything is in the cytomet. Now, once the cell culture is performed, after 3 to 4 weeks, depending on the cell maturity, you then can expose to the compound that you want to assess, and basically just take the cells, the chip, you take off the protection lead, and you place it into the Maestro system, as you can see in here. That is very easy to use. You place it, the environment is totally controlled, and after you can launch the software that we have developed so that you can record electrical activity. So here's a live demo of the software. You can see is a NetTree uplink here, directly installable onto your favorite computer, connect to the Maestro. We scan a sample QR code, or you enter the sample you want to assess. You place the NetTree plate into the Maestro system that we have just shown, and then we are ready for testing. Usually we record the recording up to 10 minutes. Here's been a shorter video, but you can see the interaction of the component with the neural network activity directly in live. And if we zoom into the different chips, you can see the electrical activity burst, etc. That makes the digital signature. We then extract the metrics and upload link to our data link system. And as an example here, we have tested several types of one specific component to assess neurotoxicity. And by our machine learning system, we're able to measure in 42 dimensions the distance between the digital signature of the compound and the one that are already into our database. And again, this is a 2D Z projection plot that allows us to make assessment. You can also have different visualization with example that we know of phosphorus compound that has a specific effect. For example, buprenorphine can induce seizure or motor deficit consequence with 3-nitropropylenic acid. And each individual dots represent a compound that has been placed and recorded into our digital library. So the more compound we have, the more accurate the prediction can be. And in the future, we also will like to develop another AI, generative AI, that allows us to, if the compound is into a specific area, what you could add to make the compound go back into a non-neurotoxic area. Well, that was the live demo. Thank you very much for your time. It was quite quick, but make it very efficient. And I'll be more than happy to answer your question if you have any. Thank you very much for your attention, and I wish you a very end of the week. Okay, so I have a question about the reproducibility of our experiments. So indeed, Reproducibility is assessed into our devices by making a lot of different replicates. Since it's a high throughput system, we are able to make a lot of them in the same experiment. So it doesn't really cost much money to make multiple stack of them. So we look at, we do a lot of replicates. And also, as I mentioned before, you are able to assess the quality control of the cells before application of the compound. So you really have an in situ quality control assessment of the cells, the cell culture itself. But also we systematically do, at the end of the experiment, we do KCl exposure to look at that the cells is actually living, and so they will be living if they have a response in the quality control, but even not destroyed by the component that we have tested. So we, let's say, have an in situ quality control over time. Okay, another question about cell sources. So indeed, we have a couple of cell suppliers that has been qualified into our premium programs. Cell supplier, let's say today we have up to 7 different type of suppliers of different type of neurons, but also non-neural cells type, because we also can place astrocytes and other different type of cells in co-culture. So we do also co-development with our customers. Non-neuronal cell type can also be placed. We have, for example, our innovative skin model that is under development, and also our innovative gut model and innovative cardiac model that is possible. And we are very open to new collaboration with cell suppliers if you have some idea and some new possibility to have digital signature of different organs. So another question, indeed, we have, in this particular demo that I wanted to show about the usability of the system and the UX that we have developed, we have, and the example was neurotoxic that was presented to the CES Las Vegas, but indeed we have different type of applications, especially in chemotherapy-induced pain neuropathies, or pain in general, vaccine pain, and anti-nausea pain, so emetic response. We also address into the neurotoxicity and, generally speaking, pesticide assessment and into food safety application. We also have some capabilities into the dermocosmetics area for eczema, diabetes, and purite and dermatitis atopic, using specific juice of cells or our inverted skin model. Yes, so we are planning to have different conference over the year. The question is, are we planning to show this live demo at the different conference? We are going to Bio Spring in Milano in March. We will have probably a live demo in a Bio International in Boston also, and obviously at MPS World Summit in June in Brussels, where you can see the demo live. Okay, so there's no more question. Again, thank you very much for your time and for your questions. Our team is here to continue the discussion if you'd like, by mail at contact@netri.com or directly to your distributors throughout the world. Many thanks. Have a good end of the week. Bye-bye.